Thanks for inviting me. This is a great place and I'm glad to be here. I'll talk today on single molecule studies of membrane structure in live cells. And that's the Gloria Castle in the background. The university is not quite as nice looking. So. What I'm interested in is the organization of the plasma membrane of living cells. And I show a couple of cells here. The plasma membrane is not only a structural component in the sense that it does delineate the inside from the outside of the cell, but it also contains many interesting structures, such as clathrin-coated pits, caveoli, possibly lipid rafts. And if the cell wants to talk to the rest of the world, it has to assemble molecules together in order to communicate. And then on a more global scale, there's a larger organization in that some cell types have a polarity in which one end of the cell may have different molecules than the other end. In the case of a neuron, you have the input down in the dendrites and you have an output at the axon. So you have to have a way of keeping the molecules here different than the molecules here. And this is the classic image of the plasma membrane, stolen from a Brescher article in Scientific American, that illustrates the fluid mosaic model of Singer and Nicholson, where the lipid layer with its embedded cholesterol is a fluid interface. And in this sea of lipids, the proteins tend to float around relatively freely. There's two things missing from a sketch like this that I'd like to bring out. One is that beneath the membrane is the actin cytoskeleton. And the actin cytoskeleton can come close enough to interact with these proteins, therefore give a means for controlling what the proteins do. And second, in a static image like this one up here, it's not obvious that everything is dynamic. The lipids are moving, the proteins are moving, the cytoskeleton is moving, and on a more global scale, the whole bilayer is fluctuating as well on top of this structure. So if we want to look at structure and what the proteins see when they walk through here, we want to watch one of these guys. Of course, you can't see a protein directly, so we have to label it some way. So we use two labeling techniques in our group. One is single particle tracking, where you take a 40 nanometer colloidal gold particle and then attach it to a protein through an antibody or ligand. The other technique is single fluorophore video imaging, where you can label a protein by a fluorescent tag like size 3, or you can attach a GFP to it and watch the GFP molecule walk around. The SPT has the advantage that this is a very easy thing to see, the 40 nanometer gold. So this high signal to noise ratio can allow for high speed imaging of the motion of the protein underneath. It has the great disadvantage that the gold is huge compared to a protein. So the chance of cross-linking 2 or 3 molecules underneath is large. So, single molecule, maybe. Whereas when you go to fluorophore, you can tell if you've got at least a single fluorophore because you can watch the one-step bleaching process. If it goes from on to off, you've got a single fluorophore. Disadvantage is you've got low signal to noise, which restricts us from doing very high-speed imaging. So for the first part of this talk, let me deal with the gold. Then I'll try to convince you the gold really doesn't do anything wrong in doesn't affect the data and what I'm talking about. So this is watching the transferrin receptor on an NRK cell in real time. There's the gold walking around. Scale bar is 1 micron. And it looks like it undergoes a random walk across the cell surface. And I'll overlay the trajectory next here. Here we go. And now it looks a little bit less Brownian. It tends to stay in certain regions a little bit longer than other regions, with infrequent transitions from one region to the next. That's better. And we find that most proteins, in fact all that we've looked at, tend to do this hop-type motion, where they stay in one area and then make a transition to another area, transition to another area, and do long time motion by doing this. And I should mention that we always show trajectories that look like they're going linearly in one direction. That's only because we choose ones that tend to do this because it's easier to look at. A lot of them will walk amongst themselves and back over themselves, but it gets muddy on the screen. So there's not a directed motion here. It's the choice of trajectory. So how do you analyze one of these? Because I hope that some people at least are thinking that this is probably junk. That a random motion can give you this. That as long as you're looking at one trajectory, pure random Brownian motion could look like that. So how do we analyze it to say that it's actually not random Brownian motion? If we look at the mean squared displacement along one of these trajectories, we get a plot like these black dots here. And characteristic of these type trajectories is that there's two linear regimes. At the short time, you get a linear regime that's got a high diffusion coefficient. And at the long times, you get a linear regime with a slow diffusion coefficient. What these are, this diffusion coefficient is indicative of the really fast motion inside of one of these areas. And then the slower one tells you how fast it walks from one compartment to the next for the long time motion. So we fit something like this to a theory for diffusion through equally spaced semi-permeable barriers, the approximation that we shown down here. And this can give us an estimate of the compartment size that we see here. We can also, the coloring of these is done through a program that we developed that takes the initial trajectory in time, makes a window of n frames, walks the window through the data, and measures a effective diffusion coefficient, and then varies the window size. What happens is, whenever you get the window such that it crosses two compartments, the diffusion coefficient is larger. As you get near the edge of the compartment, the diffusion coefficient drops, and then as you jump to the next compartment, it increases again. So what you end up getting are these lines that show up here where you get a sharp jump in the diffusion coefficient. And we use these lines to flag the points where we change the color, and then we can compare the domain sizes we find here to what we get from the average fit of the mean squared displacement plots. So what do we see? In NRK cells, done at 30 frames per second, we get trajectories that look like this. And this is actually another protein, CD44, as opposed to the transferrin receptor I showed earlier, showing the same type of trajectory. The average size of these compartments is 660 nanometers. The average residency time, how long it stays in one of these compartments, is 1.8 seconds. The diffusion coefficient inside of one of these is about 1 micron squared per second. And we find that 77% of the trajectories we see are statistically different than simple Brownian motion. So we classify them as this hop motion. We also find that if we look at transferrin receptor and CD44 on NRK cells, they have about the same compartment size and about the same residency time. That brought us to the membrane skeleton fence model, where this type of trajectory we find is due to the actin membrane cytoskeleton that's in close proximity to the lipid bilayer. This was shown through doing drug treatments to the actin and showing that that's the key component in this. And so in this type of model, the cytoplasmic portion of the protein runs into the actin skeleton and can only make it from one compartment to the next when one of sort of three things happens. The actin can break. If the actin breaks, you open a gate and things can flow through. There's no barrier anymore. Or the membrane to skeleton distance can fluctuate. Since the membrane's always fluctuating, you can have enough space that the cytoplasmic portion gets through. And then getting hit by thermal impulses, you can just happen to have enough energy that pushes it across when it gets to the edge. So if we see these in the pure diffusion of a protein, can we image them? Optically, this is a problem. If it's really the cytoplasmic portion that's running into the actin, we're looking at maybe 5 nanometers below the bilayer. So optical sectioning always will see a much deeper depth, and what you'll end up doing if you do any type of fluorescence measurement, the bulk actin will blur everything out. So in live cells, you can go to electron microscopy, but in live cells, we used optical force microscopy. Since we already have this handle up here, we can grab that in an optical trap and then scan the cell underneath. So the optical trapping laser is a 1064 nanometer YVO for laser, and we have a piezo scanner on the stage that's synchronized up to our video imaging, so we can build up images of this membrane skeleton. We're using CD44 to do this. The key point of CD44 is it has a 72 amino acid cytoplasmic domain that can sense about 3 to 4 nanometers beneath the bilayer. We're also using a stem cell factor receptor, which we've engineered the cytoplasmic domain to different depths. And we're trying to now image different depths beneath the bilayer by using this probe. Now, CD44 can bind into the actin, but we're using freely diffusing ones, so this connection's not there in what we're looking at here. Basic idea is, as I said, you grab the gold and you scan the cell underneath. And the parameter that's going to be important here is how fast you load the barriers. It's the same as in a bond failure experiment, as you heard a month ago or 2 months ago from Evan Evans. That we know if you wait long enough, the particle will get to the next compartment. It has a certain residency time. So if you grab the particle and move across slow enough, it will cross anyways. It has a certain probability of doing it. But if you grab it and drag it really quickly, it catches on the edge of the barrier, and then you build up force before it gets across. So we're going to keep our eye on how fast we try to get across these barriers and what the effect is. So this is the type of imaging we get. This is a 2 by 2 micron square on the NRK cell using CD44 as the probe. The light areas are 0.2 piconewtons of resistance. The black areas are 0 piconewtons of resistance. And we can see areas that are sort of correlated at high resistance and large areas that are of 0 resistance. And if we do it another day, another cell, we again get these long lines of high resistance with areas of no resistance, which looks a lot like the trajectories we saw of CD44 on the NRK cell. And the sizes of these areas are on the order of a half a micron. Fits right nicely with what we see from the SPT. We can quantitate the force it takes to go across by taking one of these images. I've taken a line across here and drawn it out here, where we get the peak forces it takes to cross the barriers. We can take them from a bunch of images, plot them up here against the loading rate, and we see that there is a loading rate dependence, that you get higher required forces to cross the barriers at higher loading rates, which you can calculate basically the same way you do for bond failure. Look at a particle trapped between 2 barriers. Look at the mean first passage time to cross one of the barriers, which is just solving a Smolakowski equation. Then apply a force which tilts it. You can then calculate, if you now ramp that force as you go against it, the force increases with time. And this is the simplified version. There is a much longer one I have, where you get a logarithmic dependence on the rate to the strength that you find when you try cross these barriers, which of course is this straight line I see here. The spread of the data, the increase in the spread of the data is also predicted by this as well, since it's a thermally activated process. Now, is this universal? I've shown it for the NRK cell, and I've shown it for 2 proteins on the NRK cell. Done this on a lot of different cells, and yeah, we find it everywhere. NRK cells are weird. They've got 2 sizes of compartments. I'm only showing the smaller one here at 260 nanometers. There's also the half a micron one. ECV cells, about 110 size compartments. PTK2, about 42. COS1s, about 36 nanometer compartments. Residency times change in the different ones. Compartment sizes change, but qualitatively, they all have compartments from our data. Now, everything I've shown you has gold as the label. There's huge gold at the top. And the first question I always had was, what's the gold doing? You've got a huge planet on top of a protein. Isn't that affecting everything you're seeing? Can you see this compartmentalization without the use of gold label? And this is data that I just got a week before I came here. This is a single fluorophore video imaging of CD44 on NRK cells. These bright spots are single Cy3 labeled FAB antibody to CD44, and it's done at 8 milliseconds per frame on an objective type TIR, a total interaction microscope. If I take one of these trajectories, or a couple of these trajectories, here you start to see that you still get this compartmentalization, and now we're using just a single fluorophore as the probe attached to an FAB. And when we analyze these for CD44 or transferrin receptor on the NRK, and this is again looking at the larger domains, we get 750 nanometers on average for the size of the domains, exactly what we see for the gold. So it doesn't look like the gold's causing the compartmentalization, it's only reporting it. So that's proteins. What about lipids? What do lipids do? Well, we can attach the gold to a lipid through incorporating a fluorescein DOPE molecule and using anti-fluorescein, or we can incorporate directly a Cy3 DOPE, and they hop as well. These are trajectories of DOPE on NRK cells done at 25 microseconds per frame. This is using the gold, so we can go to these extreme time resolutions. The compartment size is for the smaller compartments on NRK is 230 nanometers. Residency time is 11 milliseconds, and the diffusion coefficient inside of one of these domains is 5 microns squared per second. This is interesting. This number here is about what you expect for a lipid on a liposome, on a vesicle, about 5 to 9 microns squared per second. Yet in cells, FRAP data, recovery after photobleaching data, has always found lipid diffusion to be much slower. So what we find is that lipid diffusion on long times is not slow because the lipid is per se slow, but that it has these slow hops. And that once you get inside the smallest compartment, it's moving as fast as it is in a liposome. As basically expected. Another thing to note is that the residency time here is only 11 milliseconds. If you do this at 30 frames per second, then this trajectory is 2 spots, one there and one there. So there's no way you can see the compartmentalization unless you have high time resolution. And lipid diffusion, hop diffusion, is also universal. The compartment size reported by DOPE or protein is the same in each cell type. The only thing that differs is that the residency time of the transferrin or protein is generally higher than that of DOPE or of lipid. You'll note it's not always higher. And I can explain that in a question if anybody asks. So how do the lipids sense the membrane skeleton? The lipids are on the outside, the membrane skeleton's on the inside. So we propose this anchored protein picket model where your actin cytoskeleton has proteins that are stuck to it, transmembrane proteins that then protrude across the bilayer, and a lipid on the outside then senses these proteins and has trouble walking from one compartment to the next because it has to go between these guys. And then between them you have, you have steric hindrance, and between them you have a hydrodynamic like friction or a higher packing, a sort of a free area model for diffusion that'll slow this lipid's chance of getting from one side to the next. So it tends to keep the lipid inside of here. And since the proteins are lined up on the skeleton, they faithfully represent the skeleton mesh that's underneath. So we looked if this made any sense. This is a Monte Carlo simulations of this anchored protein picket model. Monte Carlo simulations can never tell you anything's true, but they can help you throw stuff out if it's really bad. So this was just to verify that at least it's possible. Where we did a simulation, putting a bunch of disks down in 2D, starting a point particle, letting it walk, and then putting a reduction in the diffusion coefficient around each of these posts, as explained in Almeida et al. In '92, where you have a reduction in the free area around the post. And if we do this, as we add more and more protein to the barrier, so we start filling it up, and we analyze the trajectories just like it was a real experiment, we find that it looks like it's simple when you don't have enough protein, and that at a certain point there's a transition where now you find Hopf diffusion. So the lipid, or the point particle in this sense, tends to look like it's now confined. If we run this for what we expect to see for NRK cells, ECV cells, and FRSK cells, we find that if we calculate the residency time inside of these type compartments, we find that you need about 15 to 20% of the barrier filled with proteins in order to recover the experimental values in any of these experiments. This amounts to only about 5% of the total protein on the cell. So it's not like we're taking all the protein and filling up the barriers. It's a very small amount. So we think it's a possible explanation for this Hopf diffusion, a lipid reporting the cytoskeleton meshwork. Now, in the one of the last parts, I did want to bring up some work I've just finished recently on limits of the interpretation of single particle molecule tracking experiments. There's a lot of literature now. Single molecule experiments are becoming very hip lately. So you see a lot of publications lately using this, and interpreting the results is not always simple. And I wanted to bring up one of the problems of trying to view these diffusion of molecules in the membrane. We're looking at very short residency times here. So we're looking at particles or lipids or proteins staying inside of one of these compartments for, tens of milliseconds only. And some cases here less than a millisecond. And we're looking at length scales here that range from 40 nanometers to 260 nanometers. So we're looking at very short length scales and very short times. So we sort of have a few scales that are important here. One being the compartment size, one being the rate at which it hops from one compartment to the next, one being the diffusion coefficient inside of here, and then the last important one is the observation rate. What's the exposure time on the camera in between each frame? And I'll assume that the observation rate and the exposure rate is the same, although you can vary them independently. So what happens? These were done on a Monte Carlo simulation as well. If you have simple Brownian diffusion, no problem. It's random diffusion, doesn't matter what rate you look at it. You get the same diffusion coefficient if you look at it at 25 microseconds, or if you look at it at 33 milliseconds, you still get the same diffusion, as long as it's free, simple Brownian diffusion. If you confine the diffusion into a small area, what happens? Now, these are areas of 240 nanometers, 120, and 42, estimating these types of cells. As you go to slower frame rates, you start to localize the particle in the middle, because as the protein is moving, it bounces off the wall, bounces off the wall, bounces off the wall during a single frame. So you tend to choose the position of the particle near the center all the time. So what happens is, as you look at smaller length scales, the particle looks stationary. And if you try to determine the diffusion coefficient at very short times, it dies as you get to slow frame rates. So you start to look like the particles are absolutely frozen, and they may actually be in a small domain. And I can show you this in a video. This is a 45 nanometer compartment simulation. This particle here is played back at 33 milliseconds per frame and includes the averaging at the camera. And I'll leave the hand in the middle. It jiggles a bit, but not a lot. It looks essentially stationary in the simulation. It is running, trust me. If I go to this one, this is what we'd see if we did it at 25 microseconds per frame. You can now obviously tell that particle is moving around a little compartment. And that's the same simulation. I took these frames and added them up to make this movie here. And this is where the interpretation problem comes in. If you have a particle trapped in a 120 nanometer domain, and you look at it at 25 microseconds in black, or 2 milliseconds in red, or 33 milliseconds in blue, and these are the positions that I find in the tracking routine, try to build a probability density and then make an effective potential that's holding this particle. If you do it at a slow frame rate, you find a quadratic potential, and you start to say it looks like it's being held by a spring. And if you do it at a high enough time resolution, you say, oh no, wait, it's at a square well potential. And there's been some publications recently that done at 33 milliseconds that have said that they have a quadratic potential, and I don't think you can say that with the data. You have to change the frame rate to see if this does not change. Once you get a stable potential, you're done. That's the correct potential. But the limitations due to insufficient time resolution can bring up artifacts in the interpretation. Now, if you've got hop diffusion, this is when this next parameter of the hop rate comes in, where if you're looking at it at very high time resolution, you see the hops. If you look at it at low time resolution, it now looks like free Brownian motion. And if you try to determine the short time diffusion coefficient, it looks simple Brownian at very high time resolutions, because you're inside of here. It looks simple Brownian at very slow time resolutions, because now you're looking at this, which is simple Brownian again. And in the middle, you've got an anomalous regime that just looks like an anomalous diffusion if you don't actually look at the trajectories directly. And we can look at this anomalous diffusion regime. If we look at the mean squared displacement over 4t in a log versus a log of time, then simple Brownian motion should be a flat line, and anomalous diffusion shows up as a slope. And we go from a flat line to an anomalous to a simple region. And the crossovers occur at the average time it takes to sense the barriers for the protein inside the compartment. And the second crossover occurs at the average residency time. So if your frame rate is slower than this average residency time, you'll never see the compartmentalization. And you want to be sort of in a regime like this so you can start seeing the hop diffusion. Is that real? Yeah, we see that. This is done on PTK2 cells, done at 33 milliseconds, 2 milliseconds, 220 microseconds, and 25 microseconds per frame. We see the hop diffusion at the 25 microseconds, and as we go up to 33 milliseconds, for either protein or lipid, it looks perfectly simple motion. And if we analyze these, we go from simple, classifying everything as simple at least for lipid, at 33 milliseconds, to classifying everything as hop at the 25 microsecond time resolution. And generally, we need to have about 40 points inside of here before we can statistically say that it's different than random Brownian motion. So I've shown you that the membrane skeleton can control the proteins and the lipids, or at least slow them down. The big question then is why? Is this useful? Does this actually do this in any way, for any reason? I don't know if I can say what the cell's reasons are, but we've got a couple of ideas of where this can be used by the cell in order to do useful things. One is in signal localization. When a protein's walking around on the surface, it's got basically hop diffusion, but pretty quick and long-range hop diffusion. If a ligand comes and it dimerizes, that'll slow it down because now the dimer's got to cross the compartments. If you now start getting a signaling complex with different molecules from the inside showing up, this large complex will become arrested inside of one domain. And now it can't go anywhere, and now you can signal down here to say the food was over here, without it diffusing far enough away so that it can't figure out where it got the signal to begin with. And we've seen this with the EGF receptor on L cells, where we labeled them with fluorophore, and if we looked at single fluorophores, they moved pretty quickly. If we looked at ones that were double the intensity, they moved much slower, and as we looked at triple, they were basically stationary, given the time resolution problems. Yeah.
How do you control the number of fluorophores? Because if you use F.A.B. or antibody, you forcibly label them, you get variation of 1, 2, 3, 4.
We try to keep it as low as possible, and yeah, there's a problem here that this could be not a dimer of the EGF receptor, but a dimer of the, or 2 fluorophores on 1 F.A.B. that's stuck to 1 EGF receptor. Our average labeling on the F.A.B. is less than 1. So there's a slim chance of getting 2, and there's a larger amount of dimers than we'd expect from the distribution of fluorophores. But we can't be absolutely sure on that. And this is another area where this model has, I think, explained something very well. Looking at polarity, maintenance of polarity at the initial segment in a neuron. This is a neuron cell. This is the axon. This is the cell body. There's an area here called the initial segment that divides these two parts. And the neuron wants to keep certain molecules in this end and certain molecules in this end and not really mix them. So actually, Chieko Nakata in our group was doing this work, and she stained the axon for ankyrin G, found there was a large portion of ankyrin G right at the initial segment. She also stained for sodium channels. Sodium channels are also concentrated at the initial segment, and stained for actin, and actin is also concentrated at the initial segment. So it looks like, and she's actually done now, I think, looking at the diffusion of the sodium channels in the initial segment, and they're basically stationary. So we think they're attached to the actin at the initial segment through ankyrin G. And then she looked at the diffusion of a lipid along different parts of the axon in the cell body. And what she finds is that near the initial segment, the lipid is almost stationary, 500 times slower than what it is at the distal end of the axon or in the cell body. So if we're getting a whole bunch of proteins that are attaching to the cytoskeleton and a high density of cytoskeleton right at this initial segment, then we've got a sort of a picket model here that's going to slow everything down from going from this side to this side, so that things may enter and they may just walk back out this way instead of trying to cross the long way there. So we end up having a barrier that stops things from over here from getting to here easily. And I think I'll stop there. This is the group in Nagoya. That's Aki Kusumi, the big professor in the group. There you go. And the guys I've labeled off here are the ones that have done a lot of the work with gold and fluorophore in the group, and they're invaluable as a resource. So I'll stop there. Thank you for your attention.
What happens to your parameters from the compartments if you start messing around with membrane fluidity, like adding cholesterol or messing up with the polymerization of the cytoskeleton, things that, maybe matriculating A, or things that affect low nuclease activity, things like that?
I may find the slide, but I'll tell you in case I don't find the slide. Looking at the lipid diffusion and seeing the compartmentalization with the lipid, Methyl beta cyclodextran treatment to remove cholesterol, no effect. Trypsinizing the top surface of the cell to remove some of the ECM, no effect. Latrunculin A, which depolymerizes actin, makes the compartments about 1.4 times larger. Jasplikinolide, which stabilizes actin, keeps the compartment size the same, but increases the residency time by about 2. And that's where we came to the point that it's probably due to the actin cytoskeleton. And we've done that with protein and everything, and it's pretty consistent along everything we've done. That's also true with single GPI anchored proteins. The cholesterol depletion does not affect it, which is amazing, I think.
Steve, so what about the lifetime and dynamics of the cytoskeleton? Is it essentially static obviously over seconds, but over the lifetime of the cells?
No, I don't believe so. The imaging of it, where I've done some imaging where I've done one line, there's a dynamics to it, I think, a slow dynamics, but it is moving around because the barriers tend to shift over time. There's a limit to how long I can do that because you tend to get unlucky and the particle will fall out sooner or later. Also, the one point I had mentioned earlier, and if I bring it up here, this curious bit here, that if the proteins are hitting the actin cytoskeleton and the lipids are just feeling the pickets, you'd expect proteins to always stay longer in a compartment than the lipids do. And we find in many cell types it's basically the same residency time, which had us puzzled for a bit. What we think is happening now is this is slight breaking of the actin and rebinding. So that if you break the actin, now both the molecules can make it to the other side at the same rate. So if the breaking of actin is the rate limiting step, then they should both transit at about the same rate. This would imply, though, that the breaking of the actin is very dynamic, because this is a millisecond average residency time inside of a compartment, so it would have to be on the order of a millisecond that the actin breaks and rebinds each time.
So this raises a question I wasn't going to ask it, but now I will, about your simulations. When you say something like 15% occupancy of your barriers,
right.
Does it matter whether the barriers are, in your diagram you showed them as being uniformly spaced? If you randomly space them so you have big holes in which proteins and lipids can move, you have with the same coverage, would that affect your dynamics?
It doesn't affect it on the long range. It does on the short range. What happens is some domains don't show up as well because it can go out easily, and some domains show up better. But on average, it turns out to be about the same. What's different is if you allow a mobility to all of the stuck proteins. Then you get constant opening of possibilities. Then you basically lose the compartmentalization.
So can you say which of those three options you think will closely approximate what's going on in the cell?
What I think is going on is that the proteins are sticking only long enough so that it looks immobile to the moving lipid. So if it's a millisecond here, they only have to stick for a little bit more than a millisecond, and they're stationary to the mobile lipid. So when I say immobilized proteins, I don't mean they have to be there for days. They only have to be there for the order of milliseconds to tens of milliseconds. And I think that's probably what's going on. And many people have said, why don't we see rows of proteins then on a cell that would tell you exactly the Pickett model? But I don't think they're that stationary. They're quite dynamic. They're only stationary on this sort of time scale.
So I have another question. Your very opening slide, you talked about the advantage of the colloidal gold versus the fluorescence thing. Obviously, in terms of you have to be able to build up an image. You need enough photons to see it.
Right.
If you use single molecule, single photon fluorescence, you can sample, every 10 nanoseconds or 100
right.
nanoseconds. And one thing you might, in principle, do is increase your spatial resolution, the way Paul Selvin has done, where he increases his averages. Is that sort of thing, would you end up right back in the same time domain by the time you gathered enough photons, your fluorescence sampling rate would not be? I don't understand why your fluorescence sampling rate can't be faster.
Right now, it's strictly a signal-to-noise problem. We can't localize the protein At the highest we've done is 2 milliseconds for a single GFP. But the signal-to-signal noise ratio then is about 2. And you're getting right at the edge. Like this is getting, we would never publish it, it's not good enough. We can see it and we're kind of happy ourselves, but it's not good enough to get an accurate trajectory out of. So that's all that's limiting us right now, so we're waiting for the next best camera to come out, that we can go the next level up.
You said that you need only 5% of all proteins to anchor to the cytoskeleton. What about the other proteins? I can't imagine this from simulation, but probably you can just randomly arrange all proteins to have random fences.
Right.
Attached to any cytoskeleton, but just you said you need 1 millisecond. So can you just randomly have these fences, which are not attached to any physical subject?
If they're mobile, so the rest of the proteins are all mobile, supposedly, and moving around. As soon as you allow mobility, you no longer get a confinement anymore. If you have a movement through a randomly moving amount of disks, you never see a confinement anymore. What you find is it's an anomalous subdiffusion, but not a hop, not a confined diffusion.
Maybe you should explain that term. I don't know a lot of people here.
Anomalous subdiffusion?
Yeah. Let's do a show of hands. How many of you heard that term? Just wrong spot. Anyone else? Tim, I thought you had that information. Subdiffusion thing? Subdiffusion, yes. I'm so much you would raise your hand. Maybe we can try to understand it.
That works.
Anyway,
Okay, here we go. Now I get to use my neat pen on my computer. I can draw on the screen here. Normal random Brownian motion that goes around. You expect that if you take a point and see how far it goes in time, you expect that radius to grow as time directly. So as time grows, the radius grows linearly, or the area grows linearly, I should say, in time. Subdiffusion is such that r squared goes as t to some exponent. If the exponent is less than 1, then it's subdiffusion. So what you're saying then is it's not moving far enough in time. So the long steps that you can take randomly are cut out. So it tends to take only short steps. The idea that if you have a bunch of barriers in here that are moving, but you have a guy here that you're actually watching, the bunch of barriers around are subtracting out the long steps, because they can't take long steps because there's always something in the way. So you tend to look like a subdiffusion because you take out the long tail of the probability distribution. So what would happen is that when you plot r squared versus time, it looks like this for an anomalous subdiffusion, as opposed to being linear. Yeah.
You said that your data is statistically significant to say that it's Hall
Right.
versus diffusion.
Right.
Is it statistically significant to distinguish Hall from
Anomalous subdiffusion. Yeah, that's a real question. I'm actually right now doing a bunch of simulations using an anomalous subdiffusion algorithm for the Monte Carlo to prove that we can tell compartmentalized diffusion from pure anomalous subdiffusion.
if you have, say, different type of areas, different sizes, so it will look like one step, the other step, like fractal,
Right.
You know what I mean.
Right.
And in this case, maybe anomalous will describe better than just Hall when you have just one step.
The difference between sort of, I like to say pure anomalous diffusion and hop diffusion, but I'm not sure what pure anomalous diffusion is. But something that goes by really a t to the alpha. Hop diffusion should really be, can I change the killer here? Yeah, I can. Hop diffusion should really have two linear regimes in it. And the anomalous subdiffusion should be always have a curvature to it. Data's too noisy to prove that, of course. But The algorithm I'm thinking that's going to tell us if we have real hop diffusion or not is the one that chooses the boundaries between one compartment and the next, and comparing that to the results we get from here. The anomalous subdiffusion in general, from what I've looked at, and I was talking with Mike Saxton, who does a lot of work on this sort of work as well, anomalous subdiffusion will look really compact, but it looks too compact compared to the hop diffusion. The hop diffusion tends to look compact but separated in a sense, if you see what I mean. This seems to look more separated than that does. Even though they're both subdiffusion, pure anomalous subdiffusion tends to be a compact piece that moves around, and the hop is a compact piece that jumps around. So I think doing directly from the MSD, we're never going to be able to tell one from the other, but I think there's other methods we can tell that it's distinct from pure anomalous subdiffusion. But that is a real contentious point of whether we're seeing that or not.
The optical trap provides...
And it provides a proof that there is something there, yeah. But strictly from the diffusion studies, I think we can prove one from the other. This is what Webb is always bugging us that this is just pure anomalous subdiffusion. So we've got to do something about this at some point.
The optical trap was for proteins. You have results both for lipids and proteins.
Yeah. Yeah, true. The lipids is more the problem because that's the more contentious, the more troublesome theory is the lipid one.
So I have to ask the biopolitical question from the tone of voice you used early on. What is the relationship of these results to rafts?
Oh.
Are there rafts? Could rafts be accounted for by this phenomenon?
I didn't explain our raft data, but we do have data. One of the postdocs, Kenichi Suzuki, has been working on GPI-anchored proteins and the motion of GPI-anchored proteins. What he finds is that if you label the GPI-anchored protein at such a low level that you've probably got a single one, we can check it by doing both the goal and the fluorophore and show that the diffusion coefficient is the same. It looks identical to lipid. If the pickets exist, you'd expect if there's a raft around it, the raft would have to go through the pickets and therefore it would be much slower. So we think if the GPI-anchored protein at steady state, single GPI-anchored protein, has a raft, it's really small or it's very dynamic, so that it's coming together and forming and dispersing on the order of a millisecond, or that it's small enough that it's just a couple of molecules and it doesn't matter. Next, if we now take a gold and put a bunch of IgG antibody on it, so we actually cluster the GPI-anchored protein underneath. Now we get a slower diffusion because we've clustered it already, and we see it stop every so often. We get calcium signaling. If we block, if we depolarize the actin, if we block Sark family kinases, the stopping goes away. It just continues to walk now. The calcium signaling goes away as well. And if we watch things like PLC gamma, it tends to come to this area when it stops. And so does Lin comes to that area when it stops as well. So we think what's happening, our sort of general model of rafts, is that rafts at a steady state are small and unstable. When they're liganded, they tend to now form a more stabilized raft that then recruits the molecules it needs and then does the signaling at that point, and then disperses when it needs to. That way it can move around quickly, and they can come together to make a more stabilized piece at the point when they need it, and they do the signaling.